Hello everybody, so welcome to Bite Size Biology, and today we are focusing in on excretion. Now with excretion we also cover homeostasis because they kind of go hand in hand. The main questions that you're going to be asked is to draw label diagrams of either the kidney, the nephron or the skin, to describe the effects of excretion, that's throughout the nephron itself, to distinguish between excretion and egestion, to state the importance of homeostasis, to obviously link that to ADH, the hormone. Any definitions, and there's a few there. I suppose excretion and egestion fall under that too. And also obviously there to link it to homeostasis overall, in particular for the skin as well. You'll see I have some years over the kidney, nephron and skin. So they were the last time that they came up to draw them. So the skin obviously is, and the kidney for that matter, are well overdue. But the nephron came up in 2020 and that was quite a big question. So without further ado, we'll get started there. We'll look at 2020 and we'll work our way down. So 2020, there is no marking schemes just yet. So I'm using it without the marking scheme. So I'm going to give you the answers that I have in general, but there probably is going to be other accepted answers, if that makes sense. So starting off, you're asked to explain what is meant by homeostasis, give one reason why homeostasis is important in organisms, and explain what is meant by the term excretion. Now, whenever they ask for explain what is meant by, really they're just asking you to give the definition of homeostasis. Define excretion. So let's have a look at part A there. So homeostasis is the maintenance of a constant internal environment. For instance, our bodies need to be maintained at 37 degrees Celsius for our enzymes to work efficiently. The pH in our bodies needs to be specific for our enzymes to work as well. Generally, when we ever talk about temperature or pH for the body, it always goes back to enzymes. Now, why do we need it? Well, it's there to regulate temperature, enzyme production, pH, as I said. There's a host of different answers you could say for that one. And then excretion. Excretion is the removal of metabolic waste. You can say from the body, you can say from the cells, it doesn't matter as long as you say removal of metabolic waste. That's really important that you put the word metabolic in there. Okay. Continuing on, so we're going to look at B now. Urine produced by the kidney contains water, salts and urea. State the precise location in the body where urea is made. And also, name the food biomolecule from which urea is derived from. Okay, urea is made in the liver, guys, and it's derived from protein. That's just learning off. And it's obviously excreted through that of urine. Urea, urine, you can see the connection. Part 3, and I've written it in here for the answers. You're given a diagram of the human kidney as shown on the right, and you're asked for 1, 2 and 3 there to state the location of where this function is being carried out. Now, in your state exams, you obviously cannot be putting the answers on here. Okay, it's going to be a long question. You don't have an A4 page in front of you that you'll be writing on. Okay, so I just put it here just because it's easier to connect the two of them together. So in which part of the kidney is the Bowman's capsule located? It's located in the cortex, the outermost region. In which part of the kidney is the loop of Henle located? This is located in the medulla, which is kind of on the inner regions. And you're asked to name part A. A there, everybody, is the ureter, which are connected to the bladder. IV asks you to draw a large label diagram of the nephron, and you're supposed to put in these labels here. So the PCT there, the DCT, the glomerulus, loop of Henle, collecting duct, and Bowman's capsule. Now, this is the one that I have here in the notes, so it's the one I'm going to continue on. You didn't need all of the capillaries, and I have extra stuff there in terms of what's actually happening. But you can see over here we have a glomerulus at the top, which is nesting inside the Bowman's capsule. The Bowman's capsule is connected to the PCT, connected to the loop of Henle, connected to the DCT, connected to the collecting duct. Now there's different variations of how you could draw that, but you need to have it in that system. Glomerulus, Bowman's capsule, PCT, loop of Henle, DCT, collecting duct. And that should be all of them there. It is. Okay, and that was all you needed to put in there. It's quite a nice diagram to draw, really. It's not the most difficult ones, even though it looks it. But if you take it step by step, it's actually okay. Now, C was a lovely question, only because it has never really been properly asked so much before. They've compared blood plasma and glomerular filtrate before, or they've compared urine and blood plasma, but they've never compared all three of them together, and that's including the glomerular filtrate. So it's a very neat question. It's something you can't learn off in terms of the values here. You're not expected to, but the reasoning behind these values you are expected to know. So from part I there, using the data from the table above, what is the main difference between the composition of blood plasma and the glomerular filtrate? Well, I suppose if you're looking at it, it's fairly obvious that protein is the main difference. Okay, and you're going to be asked why. Well, the reason is proteins are too large to fit through the glomerulus, and if they cannot fit through the glomerulus, they cannot form the glomerular filtrate. So proteins have to be broken down, and unless they're broken down into their amino acids and so forth, they will not form. They will not filter in through the glomerulus into the glomerular filtrate. So for that one there, guys, I'll just show you the answers for that. Protein is far higher in plasma than glomerular filtrate. This is because proteins are too large to pass through the Bowman's capsule, or the glomerulus to be precise. Because that's the Bowman's capsule is what holds the glomerulus, and the glomerulus is what does the filtration. Using the data from the table above, in relation to glucose, explain the findings in the composition of the fluids between blood plasma and the glomerular filtrate. So glucose, you can see here, they're the same. Okay, so blood plasma and glomerular filtrate. So I'll show you my answers for that. So glucose levels are the same, as glucose is small enough that it can be filtered in through the glomerulus. So glucose is small, too small to be filtered. I'm not sure that's the correct wording for it. Is what we say is small enough. That's better. To be filtered. Next, the final one on this one, the glomerular filtrate and urine. So if we're looking at the difference here, you can see here in urine there is no glucose, and there is a much higher level of urea than there is in the glomerular filtrate. So your answer there is no glucose in urine, as it has been reabsorbed in the PCT and the DCT. Okay. And that's probably going to be enough. Okay. It may be possible that there was more asked or more expected. I won't know until there is a marking scheme attached to this. Continuing on, using your knowledge of homeostasis and excretion, state the effect of the volume of urine produced by a high salt intake and explain your answer. So if you have a high salt intake, what happens? Well, a high salt intake means you will produce less urine. The reason being here is more water must be reabsorbed to ensure the blood plasma isn't too salty, which could have a negative impact on body cells due to osmosis. So more water must be reabsorbed in the collecting duct and the DCT to ensure that the blood plasma is not too salty. And the more water that's reabsorbed, the less urine produced, everybody. Okay, let's move on. 2018, you're asked to name the structures X and Y. So you weren't asked to draw it, but you are asked to label it. So X, everybody, is your Bowman's capsule. And Y, which is this part over here, is your DCT. your distal convoluted tubule. DCT would be is appropriate enough to say that. For B, you're asked to draw a line from the target area box, that's this part over here, to the precise location in the diagram to indicate whether hormone ADH has its effect. Now, two things to note about this. The first thing is the amount of students who skipped this question here in exams is scary. Just because there's no lines there doesn't mean there cannot be a question asked. So please, please, please take your time when answering. I know in exam situations it's very easy to, you want to fly through the questions as quick as possible to be finished. So B, anyhow, you're asked to locate where ADH has its effect. Well, it has its effect in two areas. You only needed to state one area for the exam. So either the DCT or the collecting duct. You did not need to name them. Now, explain what happens due to ADH on the target areas. Well, it makes the DCT in the collecting duct more permeable, which means a greater reabsorption of water. And you could write down there, less urine produced. Okay. What change in the blood composition triggers the secretion of ADH? Well, there's two ways this can happen. Either you have a high salt levels in the blood, or you have low water concentration levels. So there's two ways the blood composition triggers the secretion of ADH. High salt concentrations or low water concentrations. Now, you're asked to name the endocrine gland in which ADH is produced. Now, this can sometimes confuse students. Okay, the question says produced, so technically the only answer acceptable for this one would be the hypothalamus. ADH is produced in the hypothalamus. It is stored in the pituitary gland and released from there, secreted from there. Now, the marking scheme did allow for the 2018 one, the pituitary gland, but technically that is incorrect. Okay, so the endocrine gland that ADH is produced really is the hypothalamus. And just so you know, for future reference, for future marking schemes, I should say, you might not be allowed to say the pituitary gland for where it's been produced. If it asks where is it secreted from, the pituitary gland. Suggest one possible treatment for kidney failure, dialysis, one of them. You could also say the likes of a kidney transplant. Okay, moving on. You're going to see a lot of the questions from now on are going to be very repetitive, so we'll fly through them. 2016 question 12, you had a full question there on the likes of homeostasis and excretion. So part I, distinguish between terms excretion and egestion. And so that's you just define both of them and suggest how excretion occurs in a simple organism, that's a unicellular organism now, amoeba. So everybody, excretion is the removal of metabolic waste. Egestion is the removal of undigested waste. So that's such as feces. You have to know those definitions inside out, really. And then for amoeba, how do they carry out excretion? Well, they undergo osmoregulation, and they do that by using a contractile vacuole. The contractile vacuole sucks in the water and it expels the water then when it's full to prevent the cell from bursting. Next. Name two excretionary products other than water of mammals, and for each product, give a location in the body where it is produced. So I said urea and carbon dioxide. I said urea is produced in the liver, okay, due to breakdown of proteins, and carbon dioxide is produced in the body cells. Respiration is what takes place there. Water wasn't allowed because obviously they said water here, okay. But if you were just to state two excretionary products, you could say water, and again that's produced in the body cells. Okay, for part 3, describe the role of ADH vasopressin in human excretion. Now they're asking for description here, so you don't know what the marking scheme is going to be like, so you have to really write as much as possible for this. It's the only one out of all of B here that has a description. The rest of us is naming. So you can be guaranteed that out of the 27 marks present, most of it will be going for the description. Okay, so what is happening? So if you have a high salt levels or low water concentrations, okay, ADH is produced by the hypothalamus in response to this. The pituitary gland releases ADH, which then travels to the DCT and collecting duct, and it travels by the means of blood. It makes the collecting duct and the DCT more permeable, meaning more water is reabsorbed. And you could write down less urine produced. Now that was enough for the marking scheme, but just to put it all together. So ADH produced by the hypothalamus, stored in the pituitary gland. The pituitary gland releases this once it detects that the blood levels are too salty, or there's not enough water there. It travels to the nephron. In particular, it travels to the DCT and the collecting duct. It makes them more permeable, more water is reabsorbed, less urine is produced. Part IV suggests two structures in flowering plants which play a role in excretion. Now, this links in with the plant chapter. You may or may not have done it by now, but they are the stomata, which are located in the leaves, and the lenticels, which are located in the stem. It's likely you would have heard the stomata from Junicert, or Junicycle, but even if you haven't heard of either of those two, you will come across both of them again in the plant structure or plant transport. Okay, the final part of this question is, and we've seen these questions already, okay, for some of them, what is meant by the term homeostasis? Okay, so very quickly, homeostasis is the maintenance of a constant internal environment. And then you're given the rest of them to answer. What is the effect of an increase in metabolic rate in response to falling external temperatures? External temperatures just means your environment is getting cooler. So what happens to what is the effect of the increase in metabolic rate? Did I skip a question? I think I did. I did, yeah. I skipped a question there. Explain the term metabolism. Metabolism is the sum of all chemical reactions in the body. So we have anabolic reactions and catabolic reactions. They all fall under the heading of metabolism. And as we were saying, what happens with increased metabolic activity? Your temperature will increase, which makes sense. Okay, because we're in a now colder area, our bodies have to remain a constant temperature internally due to homeostasis, so metabolism increases, increasing our temperatures. What term is used to describe animals that can vary their metabolic rate significantly? These are known as ectotherms. So any reptiles are ectotherms. Their metabolic rate can vary massively, and they really do rely on the environment to regulate their temperatures. Suggest how animals that cannot significantly vary their metabolic rate, that's us, we are endotherms, may respond to falling external temperatures. So what happens when temperatures get cooler? Going back to number 4. So these are animals that we're talking about here. Okay, most of them will move to a warm place or they will hibernate. So, in winter and stuff like that, they hibernate, or they do not hibernate, or if it's more of a minor inconvenience, they simply move to a warm place. Okay. What is meant by the term piloerection? And explain how piloerection can be an important response to the falling external temperatures. Now, this has come up once or twice. So piloerection is simply the hairs standing up, and when they do so, they trap a layer of air close to the skin. And I need you guys to state that these are that when it traps air close to the skin, it's a really good insulator. Remember, if you look at your double glazed or triple glazed windows, they have air in between the panes of glass. the reason they have air is because air is a good insulator. It will help us keep us warm. So piloerection, and why it's important, what it is hair standing up, and why it's important is it traps a layer of air close to the skin, and it's a really good insulator. Next, explain how vasoconstriction can be an important response to the falling external temperatures. So it's getting cooler again. Constriction is like, think of a boa constrictor snake, where it gets tighter as such. It wraps around its body around the prey and squeezes to get tighter. So vasoconstriction in respect to excretion, okay, it reduces the blood flow to the skin. And by doing that, it reduces heat loss. Sometimes you might notice when we exercise, we see our faces get very red. Well, what's happening here is vasodilation, where the blood vessels are increasing in size, which is increasing the amount of blood going towards the skin to increase the heat loss due to us exercising. Now we'll go back up here. So this will conclude part 1 of excretion, because it's quite a heavy chapter, so I'm going to divide this up into 2 parts. But you can see already we've seen the bulk of 1 to 6 here, and part 2 will simply be repeating a lot of what we've been doing in part 1. And yeah, that's about it really, guys. I can't think of anything else to say other than that. I will see you guys in part 2.